Hi everyone, welcome to all. This is Vidyam Academy's most expected question series for medical NEET examination. Today's topic is 12th Biology chapter number 5 molecular basis of inheritance. And into this subtopic is regulation of gene expression translation. And now it's being presented by Rajiv Patel. Once again, let's welcome to all. Let's get started. The first question, ribosomes are the molecular machines of translation. Which of the following correctly identifies the ribosomal subunits in prokaryotes and eukaryotes? Option A, prokaryot 70 years, eukaryot 80 years. Option B, prokaryot 80 years, eukaryot 70 years. Option C, both prokaryotes and eukaryotes use 80 years ribosomes with 60 years and 20 years subunits. Option D, prokaryot 70 years, eukaryot 80 years. What is the right answer for this question? Would you guess the right answer? Let's check whether your guess is right or wrong. Option A is right answer for this question. Let's see how and why. These are ribonucleoprotein complexes made up of RNA and proteins. In prokaryotes, the complete ribosome is 70 years assembled from a large 50 years subunit. So here containing 23 years rRNA, 55 rRNA and 34 proteins. And a small 30 years subunit containing 16 years rRNA and 21 proteins. In eukaryotes, the complete ribosome is 80 years assembled from a large 60 years subunit and a small 40 years subunit. S stands for a Svenberg unit, a measure of sedimentation rate during ultracentrifugation, not a direct measure of size, which is why 50 years plus 30 years is equal to 70 years, not 80 years. And 60 years plus 40 years is equal to 80 years, not 100 years. This distinction is critical because antibiotics are streptomycin and tetracycline target the 30 years subunit, while the erythromycin targets the 50 years subunit. They exploit this difference to kill bacteria selectively. That's what option A, prokaryot 70 years, eukaryot 80 years is absolutely right answer. The question which demands for option A. Let's move to question number 2. During elongation of translation, the ribosome has 3 important sites. Which of the following correctly names these 3 sites and their functions? Option A site, aminoacyl holds growing peptide chain. P site, a peptidyl holds incoming aminoacyl tRNA. E site, exit releases charged tRNA. Option B, A site holds mRNA only. P site holds incoming tRNA. E site holds the complete polypeptide during synthesis. Option C, A site, aminoacyl holds the incoming aminoacyl tRNA. P site, a peptidyl holds the tRNA carrying the growing polypeptide chain. E site exit tRNA exits after releasing its amino acid. Option D, A, P and E sites are all found only on the large ribosomal subunit and function identically in all 3 positions. What is the right answer for this question? The answer C is right answer for this question. Let's see why and how. The ribosome has 3 tRNA binding sites that work sequentially during elongation. The A aminoacyl site is where each new aminoacyl tRNA enters. The incoming tRNA brings the next amino acid to be added, the growing chain. The P, a peptidyl site, holds the tRNA that carries the growing polypeptide chain. The peptide bond is formed between the amino acid in the A site and the peptide chain in the P site, catalyzed by peptidyl transferase activity of the 23 years by 28 years rRNA. That was in the P site. Now without its amino acid moves to the E exit site and leaves the ribosome. tRNA from the A site moves to the P site carrying the elongated chain, and a new amino acyl tRNA enters the empty A site. That's what option C is right answer for this question. Next, question number 3. Transfer rRNA tRNA is called the adapter molecule of translation. Which feature makes it an adapter and who first proposed this concept? Option A. tRNA carries the genetic code in its sequence and was proposed by Rosalind Franklin. Option B. tRNA directly reads the DNA template and carries amino acids proposed by Mendelssohn and Stahl. Option C. tRNA catalyzes peptide bond formation between amino acids and was proposed by Watson and Crick. Option D. tRNA has an anticodon that reads the mRNA codon at one end and an amino acid attachment site at the other, so bridging genetic code and protein. This adapter concept was proposed by Francis Crick. What is the right answer for this question? What's your guess? Let's check whether your guess is right or wrong. Option D is right answer for this question. Let's check why and how. Francis Crick proposed the adapter hypothesis in 1955. He predicted that a small adapter molecule must exist to physically link the nucleic acid language of codons to the amino acid language of proteins. Since there is no direct chemical affinity between codons and amino acids, the tRNA molecule perfectly fulfills this adapter role. Its anticodon loop. At one end, the L-shaped molecule base pairs with the complementary codon on the mRNA, while the 3-CCA end at the other end is covalently charged with specific amino acid by amino-acyl tRNA synthesis enzymes. By bridging the codon-anticodon interaction and the amino acid it carries, tRNA translates the genetic code into a sequence of amino acids, making the adapter name perfectly descriptive. That's what option D is the right answer for this question, which demands for. Let's move to question number 4. The lac operon in E. coli was the first operon described and is the classic model of gene regulation. Which option correctly lists all essential components of the lac operon? Option A. Promoter plus operator plus structural genes. Lac Z, lac Y, lac A plus a regulatory gene, a lazuli, LACL, encoding the repressor protein. Option B. Promoter plus enhancer plus structural genes and silencer, all required for lac operation expression in E. coli. Option C. Only the structural genes lac Z, lac Y and lac A. These are sufficient for lactose metabolism without any regulatory elements. Option D. Promoter plus ribosome binding site plus structural genes plus poly A signal, same as eukaryotic gene organization. What is the right answer for this question? Yes. Option A is right answer. Let's see how and why. The lac operon Jacob and Monan 1961 is organized as a cluster of functionally related genes controlled as one transcriptional unit. The promoter is where RNA polymerase binds to initiate transcription. The operator is short DNA sequence between the promoter and structural genes, is where the lac repressor binds to black transcription. The three structural genes are lac Z, encodes B galactoside is cleavage and lactose into glucose and galactose. Lac Y encodes a permease transpose lactose into the cell. And lac A encodes a transcyclase metabolizes certain sugars. Upstream of the operon lies in regulatory gene laxil, which is transcribed independently and continuously to produce the lac repressor protein. So when lactose is absent, the repressor binds the operator and blocks transcription of all three structural genes simultaneously. A beautifully efficient regulatory system. That's what option A is right answer for this question. Next, move to question number 5. In the lac operon, the structural genes are switched on when lactose is present. What exactly happens at the molecular level when lactose is available? Option A. Lactose directly binds to the promoter and recruits RNA polymerase to begin transcription. Option B. Lactose acts as a co-pressure. It binds to the repressor protein and makes it bind to the operator more tightly. Option C. Lactose is converted to alactose, which acts as an inducer by binding the repressor, causing a conformational change that makes the repressor loss affinity for the operator, freeing the structural genes for transcription. Option D. Lactose directly degrades the repressor protein, permanently switching on the lac operon genes. What is the right answer? The right answer for this question is option C. Let's see why and how is option C is right answer here. When lactose enters the cell, a small amount is converted to allactose by the residual B galactosides always present at low levels. Allolactase is the true inducer. It binds to the lac repressor protein, a tetramer encoded by the lesyl. And the causes of conformational change dramatically reduces the repressor affinity for the operator DNA sequence. The repressor falls of the operator and the RNA polymerase, which was already bound to the promoter but stalled, can now proceed to transcribe the three structural genes, LAC-Z, LAC-Y, LAC-A. This produces the enzymes needed to metabolize lactose efficiently. So when all lactose is consumed, allolactose levels drop, the repressor regains its active conformation, rebinds the operator, and transcription stops. A beautiful negative feedback regulatory loop. That's what option C is right answer for this question. Next, question number 6. In prokaryotes, the small ribosomal subunits, the TS, must find and bind the correct start codon on the mRNA. What sequence on the mRNA guides the ribosome to the correct AUG start codon? Option A. The poly A tail at the 3 end of the mRNA attracts 30S subunits to the start codon. Option B. The 5M7G cap on the mRNA is recognized by the 40S subunit, which then scans for AUG. Option C. The entire 5 UTR sequence is complemented to the 16S RNA and positions the ribosome anywhere on the mRNA. Option D. The Shane-Dalgonov sequence is purine-rich stage about 5 to 10 nucleotides upstream of the AUG base pairs with a complementary sequence on the 16S rRNA of the 30S subunit, positioning the ribosome at the start codon. What is the right answer? Let's check whether the guess is right or wrong. Option D is right answer for this question. Let's go to detailed explanation. In prokaryotes, the mRNA has no 5 cap and ribosomes do not scan from the gen. Instead, 30S subunit finds the correct AUG via the Shane-Dalgonov SD sequence, a short purine-rich consensus sequence, AGG located 5 to 10 nucleotides upstream of the start codon in the 5 UTR of the mRNA. The SD sequence is complementary to a sequence at the 3 end of the 16S rRNA in the 30S subunit. The base pair directly physically positioning the 30S subunit, so the start codon AUG falls precisely in the P site. Initiation factors F1, F2, F3 and Fmet tRNA are then recruited. This mechanism differs from eukaryotes where the 5M7G cap is recognized by F43E and 43S pre-initiation complex scans of the mRNA from the 5 end until the encounters of Kojak sequence context around the first AUG. That's what option D is right answer. Let's move to question number 7. The lac operon is regulated by both negative control, the repressor, and positive control. So catabolic repression, CAMP system. Which statement correctly describes positive control of the lac operon? Option A. Positive control means the repressor protein activates transcription by binding the operator sequence. Option B. When glucose is absent, CAMP levels fall. This initiates the CAP protein and reduces lac operon transcription. Option C. When glucose is absent, CAMP levels rise. CAMP binds CAP, a catabolic activator protein, which then binds the CAP site upstream of the lac promoter and stimulates RNA polymerase binding and transcription. Option D. Positive control only operates when both glucose and lactose are present. So CAMP independently turns on all sugar metabolizing operants and Simon. What is the right answer? Option C is right answer. Let's see why and how. The lac operon is maximal transcribed only when two conditions are met simultaneously. Lactose is present, negative control repressor removed, and glucose is absent, positive control cap. CAMP activates. Glucose inhibits adenylyl silase, keeping CAMP levels low and CAP inactive. When glucose is absent, adenylyl silase is active. CAMP accumulates and CAMP binds to CAP, a catalyte activator protein, also called CRP. CAMP receptor protein. The CAMP cap complex binds to a specific site upstream of the LAC promoter and makes direct contact with RNA polymerase, stabilizing the polymerase promoter interaction and dramatically stimulating a transcription. The positive control system ensures that E. coli uses glucose, the preferred carbon source, first. Only when glucose is exhausted does the full lack of transcription level turns on to the use next available carbon source lactose. That's what option C is right answer and rightly explained here. Question number 8. Match the column. First one is a term and second one correct description. So here the first one, Opron. Option A cluster of functionally related prokaryotic genes controlled by a single promoter and operator. Option 2, a repressor protein. Option B, tRNA enzyme that attaches the correct amino acid to its specific tRNA. Option 3, peptidyl transferase. Option C, protein that binds the operator and blocks RNA polymerase from transcribing the structural genes. Option 4, amino-acyl tRNA synthetase. Option D, ribosomal activity by rRNA that catalyzes a peptide bond formation between amino acids. Option A, 1A, 2C, 3D, 4B. Option B, 1C, 2A, 3B, 4D. Option C, 1A, 2D, 3C, 4B. And option D, 1B, 2C, 3A, 4D. What is the right answer? Option A is right answer for this question. 1A, 2C, 3D and 4B. The first one, a unit of prokaryotic gene organization where a group of functionally related structural genes are transcribed together as a single polycystic mRNA under the control of one promoter and one operator. The LAC-opron and TAP-opron are classic examples. Repressor protein 21C, a regulatory protein that recognizes and binds the operator DNA sequence. When bound, it physically blocks RNA polymerase from transcribing the downstream structural genes, constituting negative control of gene expression. So peptidyl transferase. So here the enzymatic activity of the ribosome that catalyzes formation of the peptide bond between the amino acid in the A site and the growing peptide chain in the P site. The activity resides in the 23A rRNA prokaryotes. It's intracyclic ribosome activity of the ribosome. Amino-acyl tRNA synthesize a family of enzymes, one per amino acid, that catalyze the ATP. So dependent attachment of each specific amino acids to its cognate tRNA, the charging reaction that creates amino-acyl tRNA. Let's move to question number 9. Assumption A. The TRP-opran is a repressible opran. It's normally on a transcribed continuously and is switched off when a tryptophan is present in the cell. So here the reason are, tryptophan acts as a copressor. It binds into the inactive trap apropressor protein and converts into the active repressor, which then binds the tap operator and blocks transcription of the tryptophan biosynthesis genes. So here the options. Both A and R are true and R is not the correct explanation of A. Option B, A is true but R is false. Option C, both A and R are true and R is the correct explanation of A. And option D, A is false. What is the right answer? Option C is right answer. Let's see how and why. Assertion A is absolutely right because the TAP operan, so controlling genes for a tryptophan biosynthesis, is a repressible operan that is transcribed by default when a tryptophan is caged. Because the cell needs to make a tryptophan, it is switched off when tryptophan accumulates. The cell shuts down biosynthesis when the product is already plentiful. Reason R is also true and gives the precise molecular mechanism. The TAP repressor protein is encoded by the TRPR gene and is synthesized as an inactive apropressor that cannot bind DNA alone. Tryptophan, the copressor accumulates, it binds to the apropressor and causes a conformational change that creates a functional repression. The tryptophan repressor complex binds the TRP operator, prevents RNA polymerase from transcribing the 5 TRP biosynthesis genes, or directly casually explains A. The co-pressure mechanism is the molecular reason the operand is repressible and switch off when a tryptophan is present. A, confirming option C. The final question of this topic 10. Arrange the following events of translation in correct chronological order. The first one, P, the small ribosomal subunits binds the mRNA and the Shane-Dalgano sequence prokaryotes and position at the AUG start codon. Q, initiator tRNA, fmet tRNA in prokaryotes binds the P sites of the assembled ribosome, or large ribosomal subunit joins complete 70S initiations complex formed. Yes, elongation successive amino acyl tRNA enters A site. Peptide bond forms ribosomal transcolates one codon. And T ribosome reaches a stop codon UAA, UAG, UGA. So no tRNA enters the A site. So U, release factors bind the A site. A polypeptide is released, ribosome disassembles into subunits. So here are the options. Option A, QPRSTU. Option B, PQRSTU. Option C, PRQSTU. Option D, RPQSTU. What is the right answer? Did you guess? Option B, PQRSTU is the right answer. Let's go to detailed explanation. So translation follows a three-phase sequences: initiation, elongation, and termination. P comes first. The small 30S subunit binds the mRNA, the Shane-Dalgano sequence, and locates the AUG start codon. Q follows. The initiator fmet tRNA carrying formal methionine binds the P site of the small subunit. Anticodon base pairing with the AUG codon guided by initiation factors F1 and F2, GTP, F3 even. R is next. The large 50S subunits joins the 30S mRNA fmet complex after F3 and F2 dissociate, forming the complete 70S initiation complex with the initiator tRNA in the P site and an MTA site ready for the second codon. S is the elongation phase. Each elongation cycle involves amino acyl tRNA entry to the A site, peptide bond formation by peptidyl transferase or RNA, and the translocation powered by GTP hydrolysis. This repeats for every codon. T signals termination when a stop codon appears in A site. No amino acyl tRNA can enter. U completes the process. Release factors RF1 for UAA, UAG, RF2 for UAA, UGA recognize the stop codon. Stimulate hydrolysis of the peptidyl tRNA bond, releasing the polypeptide, and then ribosome dissociated into subunits for reuse. So this is the right explanation and right sequence and chronological order which related to the translation. So here we explained 10 most important and expected questions for your upcoming NEET examinations. And next, I will come up with another topic which are very important for your NEET examination. Till so far, I would like to conclude this session with very well goodness. Thank you, and thank you so much to all.